Namaskar. Today we'll study amino acids and genetic code. Amino acids are the basic component of your protein. So, protein is a polymer of your amino acids, and in our body we need to synthesize proteins. So, we need amino acids, and which protein is to be synthesized, that is available with DNA. So, DNA is having the genetic information. It possesses all the information which protein is to be synthesized. So, DNA is transcribed into RNA, messenger RNA. DNA is transcribed to messenger RNA. The enzyme here is DNA dependent RNA polymerase, and the process is called transcription. So, the messenger RNA has the codes. As you know, this RNA is a chain of your sugar and phosphates, and each sugar is having one base, and the messenger RNA can have one of these four bases linked to each sugar. So, A, adenine, cytosine, guanine, and uracil. So, A, C, G, U. So, when we have four bases, then we can have codes in triplet 4 to the power 3, that is 64 codons we can have. And we have total 20 amino acids we have, and 20 amino acids we can divide them into essential and non-essential. Essential amino acids, why we call them essential? Because they cannot be synthesized in our body. We must obtain them from food. So, the examples are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. And non-essential because we can synthesize them in our body, and when we synthesize in sufficient quantities, they are dispensable, and when synthesis is limited under special pathophysiological conditions, for example premature babies, then we call them as conditionally essential. So, dispensable amino acids are alanine, aspartic acid, asparagine, glutamic acid, and serine. And the conditionally essential amino acids are arginine, cysteine, glycine, glutamine, proline, tyrosine, all these. So, we have four bases in our RNA, messenger RNA, that is UCAG, and if we write here first base, and in this line we write the second base, and here the third base UCAG, 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 UCAG 4 times. So, we can have first base, second base, third base, first base, second base, third base. Like this we can arrange them into 4 to the power 3, that is 64 codon we have. So, all these codon, this triplet of bases signify one amino acid. So, one codon is there, AUG, which codes for methionine amino acid, and it is called the start codon. So, almost in all proteins, methionine is the first amino acid. Similarly, there are 3 stop codons, UAA, UAG, UGA. This is stop codon, and the rest you can see for leucine. There are 6 codon, UUA, UUG, CUU, CUC, CUA, CUG, these 6 codons. For some like lysine we have 2, AAA, AAG, and for some there are 4 like your alanine, GCU, GCC, GCA, GCG. So, all these codon signify one amino acid, and this codon is present in our messenger RNA. So, the messenger RNA, the base sequence are there, and from that base sequence we can say that in triplet if we will take, so that triplet, suppose here UGC is the first codon, then similarly here there can be further codon. So, this one will signify one amino acid, and in this way we have the genetic code, and this code is available in the messenger RNA. Accordingly, transfer RNA will provide the required amino acid, and in this way this protein synthesis occurs. So this is all about your amino acid and genetic code. Thank you.